Okay, I have to resume my recording. Okay, so there you go. So we talked about this negative regulator and then positive regulator of the vulva formation, right? And I think that's, we mentioned about also LIN1 is the negative regulator of the the formation of the structure. So I think that's the loss of function of the LIN1 is the multivulva. So I think that was the last thing I think I talked about. And then I think that's the important one was, you know, after this, and then we also talk about this. I think that today is a continuation from the you know, that linear pathway, and then what we are going to more focus on today is about this, how those lateral signaling can be used for you know, different purpose. I mean, it's almost the same event, but also to be exact, this lateral you know, interaction can be used for some kind of random event. I think in here it is showing as the stochastic event. Okay. So before I begin, so do you guys have any question or something that I have to clarify, you know, before I move on? You can do so now so that I can, you know, at least make sure that people understand it. Okay, I think that you can continue. You can also ask other questions at the end of this lecture. So you can do that also. Okay, so that is, I'll just change this to a, you know, presenter view, okay? And then you can, we'll just discuss it, okay? So this is about the important decision can be random. I think that's right after those, you know, vulva formation diagram slide, isn't it? So actually, they are equal fate. So as you can see, you know, these two cells are equally giving out signals, receiving signals each other. So when they are balanced, they are, you know, same fate. They are not like a differentiated, but somehow something happened, and then they'll tip this kind of like a scale. So it creates some imbalance. So one cell give more of the ligand, and then the other cells give the you know, receive the signal. So because of that, the other side, they are losing this ability, and then slowly they are locked into that kind of ability, and then result is that these two cells undergoing you know, different cell fate. So you know, you can change. So if you are just looking at these two cells, then you can change this arrows, and then, you know, the receiver signal backward. I mean the other way around, and then it could be the same way, isn't it? So I think that's, you know, here. So you can think about this way, isn't it? And then because of that, also this cell fate can be you know, switched, but result is the same because they both have the equal potential. So sometimes this cell become green cell, sometimes it's become you know, green cell become this cell. So in that case, we can almost say 50-50 chance. So in that case, that is almost like a random. Okay, right. So let me move to a next slide. So one of the example in the C. elegans development that you can apply this kind of stochastic event is the somatic gonad progenitor cell division. So somatic gonad is the I would say ovary. Okay, the gonad that the is a somatic cell, somatic tissues, not the germ cells, egg or sperm. So those somatic gonad, you know, they are starting from this Z1 or Z4, that is the two cells. So those are the progenitor cell. And then these two cells undergo cell division like this. And then Z1 PPP. So Z1 posterior descendant, posterior, 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 okay? And then in case of Z4, that is the AAA, which is the, anterior, anterior, anterior. And these two cells, they can form either, ventral uterine cell or anchor cell. So we know that the importance of those anchor cell, isn't it? The anchor cell is the one that gives the signal to the vulvar precursor cell. In regular situation, there is a P6 cells. And then I think the Lin3 is the signal that coming from, anchor cell. So if you look at these development really carefully, and then what people found is here. So usually the 50-50 chance. So once this cell become anchor, and then this cell become VU cell. If this cell become VU cell, and then this cell will become an anchor cell. And if you screen for so many different animals, and then really is the 50-50 chance. So they look at, this is very interesting. So whether that is really depending on the these two interaction between two cells only. So they use the laser beam. So this is the laser sign. So they ablate. They call it a laser ablation. And then this is the big, L1 gonad when they are very small. And then they target this each cell, and then shoot the laser to the nuclei, and then you can burn that the nuclei in really 1/100th of a second or something. It's a high laser beam. So you kill all this somatic gonad, and then you leave the only Z1 PPP and Z4 AAA. What will happen is that, okay, these two cells still form, anchor cell and VU cell, they are still 50-50 chance. So what they did is the next, they said, okay, so they leave the other cells intact, because from this result showing that cells other than Z1 or Z4 PPP is nothing to do with this formation of the anchor cell or VU cell. So in this experiment in here, they didn't have to, actually bother with this removing other cells. Oops. Okay, so I'll just move to here, and then they just kill the Z4 cell, okay, and then just leave the Z1 cell intact, and then Z1 cell become anchor cell, and then they did to a Z1 cell, they kill the Z1 cell, and then Z4 cell become anchor cell. And then they kill the all cell, and then leave the only Z1 cell intact, and then they are still forming the anchor cell. So from this, I think they realized that, okay, so it looks like the position of the nuclei in the gonad soon after the cell is cell. Okay, just a second. Something is blocking here. You guys don't see it, but cells are born, and then looks like it's like a anchor cell is the default state, and the next that they found is, okay, they look at the this Lin12 mutant. Okay, so what they did is that, okay, so they found the Lin12 rescue animal, that the Z1 and Z4 AAA, the Lin12 is gone, okay? And then what they found is that when Lin12 is gone, then they were able to form, these, just keep on, coming back. So they were able to form, both of them were able to form the anchor cell. So, and then what they did is that, okay, next is the, they use the Lin12 mutants that are constitutively active Lin12. So this notch signaling is always on. Then Z1 PPP, and then Z4 AAA both turn into a, VU cell. So the what does that tell you? Lin12 is important for, looks like, formation of VU, isn't it? Right. Okay. So that is their finding. Okay. So next is that, okay, so they created the bias in cell fate choice. So they found the mosaic animal that the Z1 PPP is the Lin12 minus. Okay, so negative Lin12. And then all other cells are wild type, so they have all Lin12. And then when the cells, Z1 PPP is the has a lack of the Lin12, because that's why the Z1 PPP turning into an anchor cell. And I think that they did the same thing to Z4AAA. They removed the Lin12 from Z4AAA. Also, they are turning into an anchor cell. I think that's you have seen it before. Okay, so from that, they have I think that concluded that the decision to become anchor cell or VU cell is 50-50 chance. So that is the random, okay? And then the anchor cell looks like a default fate, okay? So, no matter what, they'll just leave a one cell and then they're turning into an anchor cell. So it looks like, hey, so anchor cell is the default fate. And then the is a Lin12 signaling dependent, isn't it? And then Lin12 is in fact determines the VU fate. So from this, mosaic animals, you have seen like a slide, 2 slides before. Cells that lacking Lin12 always become anchor cell, and then cells with Lin12 always become VU cell. Okay. So model is that I think that this is something going on between this only 2 cell, Z1 PPP and Z4AAA. They are, interacting together, and then they are assess. They are kind of examine. They are comparing the relative level of Lin12. Okay, and the one with a higher Lin12 become VU cell. And then we're going to see a little bit later, but feedback mechanism that ensures the only one become VU and then the other become anchor cell. Okay, so that is their observation. So they did some demonstration. Okay, so what they did is that they copied, or I think that let me use stylus here. So this is the Lin12. Can we just write it down? Lin12 promoter here. Okay, let me see. Let me see if I can do that. Okay, so this is the actually promoter. Okay, so they only clone the promoter. Okay, and then they added Lexi. If they did it in these days, then probably they use GFP or something else. But at the time, GFP was not available. Okay, and then this is like we call it a reporter construct. Okay, this DNA we call it a construct. Okay, so they made this kind of like a reporter construct, and then they inject into the animal, and then they if they stain the Lexi with the what is it they induce it, and then I think that's X scale, isn't it? If they treat the X scale, then it will create the color. It's a dark color. I think that's their the dark color is this they are representing. I think that's okay. Now I lost again. Okay, so X scale will give the this Lexi the color over there. Okay, So they made a two construct. So Lin12 LAC construct, and then LAG2 LAC construct. And then I think that's they did this kind of experiment expressing this, thing to the, animal here. Okay. And then they look at the expression of those. Okay. So let me just move. Okay. I think. Okay. Okay. All right. Okay, so mouse. Okay, so this is the next. Okay, so what they did is that, okay, so here they have a two row of the picture here, isn't it? So what it is that this is a different two different animals. So one animal has this Lin12 reporter construct, and then this has the lag2 lag-C, which is a lag2 reporter construct. So what it does is that the Lin12 lag-C, what it tells you is that it tells you the which cell is expressing Lin12. Okay, and then also in here, what it does is that it will show you which cells expressing lag2. So you can see in here, Lin12 is expressed in Z1PPP and Z4AAA in pre-forming. So it's like a earlier, very, very earlier, like a, the two cell were just born or something. Then actually the two cells, both cell, expressing the, Lin12. And also, although it's a different animal, but they have the lag2 reporter. So they look at the those Z1 PPP and then Z4 AAA, and then in early stage, also these two cell expresses lag2 also. So both cells express Lin12 and lag2 at the same time. Okay, right. And then while they are forming, and then I think that's the picture itself is not very good, but somehow they will one cell lose the activity. So only the other cell has the stronger, expression, okay, right. So, you are seeing like a diminishing effect. And when they are committed, other cell just lose their totally of the other, gene expression. And then only one cell will have the, ability to express either Lin12 or lag2. Okay, so that is the AC, and then that is the VU because there's a Lin12. Okay, so that is the their observation, and then okay, and then what had happened is that, okay, this equal two cell. I think that's maybe this is the I mean, you can say anything. Maybe this is a Z1 PPP, this is a Z4 AAA. And initially, two genes were expressed, in equally in both cells, and somehow stochastic event. So one cell facilitate the, kind of like a donor activity, and then the other cell only, settle to a receiver activity. And finally, because of that, some kind of feedback mechanism occurs. Okay, so the other ability will be turned off, and then these two cells, was determined in the in a different way. So they are undergoing different cell fate. Okay, so I'm going to move to next one. I think that this is showing you the same thing. But when you have the have a lag-Z, Lin12 lag-Z in the Lin12 mutant, they just want to show that whether lag-Z is expressed or not. So of course, this has no Lin12, so Lin12 lag-Z is not expressing. But in the same animal, if you express lag2, and then reporter, and then these two cell expresses the lag2, and then both become anchor cell. and then this is the normal one. And then this is the Lin12 is constitutively active. So Lin12 is always on. So you can see Lin12 reporter, two cells expressing here. So if you look at the lag2 in this animal, and then lag2 is not expressing here. So that's why both cells become VU cell, okay? So do we have any question? I think this is the, you know, pretty much finished with this, you know, the lecture 29. So if you have any question over this lecture 29, you can do so now. Okay, so this is the diagram, you know, I copied from the, you know, look at this. Can you believe this is, you know, this is coming from, you know, 1994, and they already find out the this technique. And then this is being a stochastic event. So a certain stochastic event that, you know, these two cell between the two cell occurs, and then one cell become, you know, Lin12 expressing cell, and then the other cell become lag2 expressing cell. Okay. So let me finish with the, you know, this lecture, lecture 29 with this slide. So you will see this two diagram here. This is not really about the Lin12 story, but, you know, this, the beautiful, I would say, such like a, you know, precise mechanism that we learn from the vulva cell induction signal in C. elegans that you see in the upper panel is the same thing, same kind of you know, signal is occurring in the Drosophila, that is the you know, eye development. I mean, Drosophila's signal is even I would say cooler than you know, C. elegans signaling, that this you know, number 8 cell, you know, finally induced number 7. So number 7 cell is really important to become a photoreceptor. I heard that that becomes like a recognizing some colors. And then when they do that, they use the same, you know, MAP kinase pathway. You know, so actually the we use the term SOS, that is the son of sevenless. That is coming from this sevenless gene, that is the receptor, EGF receptor in Drosophila. And then I think the signal coming from the RA cell is the boss, which is the bride of sevenless or something like that. And then that is coming from, you know, 8A, RA cell. And that is actually the same, you know, almost identical, you know, signaling process, just used in a different system. Okay, so if you don't have any question, then I'll just move on to our next slide. Okay, oh, no. Did I finish? No, sorry. I mean, I think I'm confused. Sorry, I'm very sorry about it. Okay. So then I'm gonna move to
from Google Chrome.
Just a second.
Trending on our college.
Just a second. I'm going to okay. So next topic is the cell cycle. So I think that the cell cycle is going to be the next topic of my, I mean, last topic of my lecture. So, you know, probably we have today and then tomorrow on Friday, and then also next week. So I think that I can complete all the lectures from this 5, you know, lecture times. And then if we have some time left, then we can go back, and then something that we didn't cover, we will, you know, talk about it. So, I mean, I assume that everybody learned about the cell cycle. You know, I teach cell cycle in 1305, and then also BIO4308 that the molecular cell biology class. In fact, I'm teaching those cell cycle and then cell division now. It's very, very complicated, but this is one of the best example that how genes are regulated, and then how those genes are really going up and down. I would say level of those gene expression activity level is controlling cell behaviors and everything, and then how cell kind of like controls the make sure that cell cycle advance to the next step. Okay, to do that they have some kind of creating some kind of surveillance system, and then how those surveillance system works, and then also how the cell maintain that the this cycle doesn't flow going backward. So to prevent the going backward, they remove the all these components at the certain. They finish their cell phase, cell cycle phases. Okay, and then also what we didn't learn, but the important thing is that there are some clear cell cycle stop. So like a cell cycle exit, and then cell cycle will be arrested for a while, and then cell cycle need to go back to the cycle if they need to, and how those things are regulated, and then how those things are related to human cancer, especially the definition of cancer is the disease of the cell cycle. So we'll just touch base of those. But the major focus on this lecture series is about the cell cycle. So something that you learned about cell division during the mitosis, how the cell chromosome condensate and then, aligned in the center and everything can be a little bit off the topic. So I mostly focus on how the cell cycle is regulated. Okay, right. So I think that's this is basic cell cycle. You have this interphase period, and then you undergo mitosis, isn't it? Right. And then cell will divide, and then they'll form these two daughter cells. So that is the basic of the cell division or cell cycle. Basically, they are forming these two cells. So we're going to learn about this that is controlled by cell cycle. So what do we mean by cell cycle? I mean, we know that, M phase, G1, S phase, G2, and then coming back to M. But we should know the individual event, a biochemical event occurs in the each phases, and then how those cell cycle is regulated, isn't it? And then, what is the concept of those checkpoints and everything is very, very important. Okay, so, cell cycle, largely we have a big interphase, okay? And then we have M phase, that is the mitosis. And the interphase, you have a G1, S, and G2, right? Okay, so we'll just review what we have now. Okay, so I appreciate you can answer. Okay, some of it. Okay, so what is the what's happening during S phase? Too easy, right? DNA replication.
Yeah.
Yeah. So how many DNA molecule do they make? How many copies do they make?
Is it just one?
Yeah, they just make one copy, isn't it? Right. So the thing is that copy material, you make sure that you only press the press the button only once. You don't want to press twice, isn't it? Right. And then so cell has to keep those kind of like a copy number, isn't it? So let's say you accidentally press the button twice, then they will stop, isn't it? So not only copying DNA is important in this S phase, but also you need to have some kind of surveillance system that if you accidentally press one more button one more time, And then what if you get an extra copy? Then what you're gonna do? And I think that is really important. Okay, so what is G2?
It's preparing the cell for division, so it's making sure that everything's kind of like in place and check, and it's still like growing the cell and preparing it for division.
Right. So, it's a preparation for mitosis, isn't it? So that is the definition. Then what is a G1 then?
Growth.
Growth. Okay. Isn't it? So let me ask you. So is growth is division, isn't it? Can we if I say, okay, cell growth is equal to cell division, am I wrong?
Not really, because it's doing organelle replication.
Right. So I think that growth will include division, isn't it? So what is it? I mean, G1. What will happen? What's happening during the G1?
It's replicating everything that's not the DNA.
Okay, not in the DNA. Okay, so yeah, I don't think it's what was what I would say is the G1 is the cell cycle arrest. Yeah, cell cycle stop. I shouldn't. I'm not going to say cell cycle exit, but that is the cell cycle arrest, or it stops the cell cycle for a while to do their own job. So let's say if we have a certain cell, maybe some kind of gland cell or something, and then if it's a gland cell, they are differentiated into gland cell, then those cells has to do their own job. Either they have to respond to the signal, or they produce some kind of hormones or saliva or something like that. And usually G1 is that kind of purpose. So they are dedicating the cell cycle for their physiological purpose or goal. Okay, right? Because they are making DNA and then dividing or something. They are too busy, so they cannot do anything else. Okay, let me put it that way. Okay, so we'll keep that in mind, and then we'll find out why I'm saying that G1 is the cell cycle arrest. Okay, right? Okay, so next is it has the different length, isn't it? So usually you have the interface. G1 is somehow in here. It's not really expressed very well, but they have this G1 phase in here, and then S, and then M phase. Looks like there's a lot of events going on, and then they have to divide, and then they are going falling into a G1 again. So if we look at this, look at the cell in a little bit, kind of like was it the in much bigger scale, then you can say, what is it? This is kind of like a mitosis, mitosis in here, so interphase, and then they are undergoing mitosis, and then cytokinesis, and then they are interphase. But the thing is that let's think about the, we'll keep this, bring this cycle to the very early, event. So when they want to form, zygote, and then they have to egg fuse to the, sperm, and then they'll have a mitosis occurs, isn't it? Right. So think about it. So at that time, right before the fertilization, and then if you look at the oocyte, isn't it? And then oocyte, and then if you go to the further back, these cell underwent what? What kind of event they went through? That is meiosis, isn't it? Isn't it? Right. So they had a meiosis for a while, okay? And then meiosis is almost ready, and then when the sperm enters, okay, and then they are changing into mitosis, isn't it? So that is some kind of continuation, isn't it? So, and then what is the important point is that it's the same cell, isn't it? Zygote, I mean the oocyte, is just accept sperm and then they change the mode of cell division, isn't it? So the thing is that what it means is, okay, any cell is the same cell. They have ability to do meiosis and then mitosis, isn't it? Right. And then what is it? Right after the fertilized, okay? And then what will happen? So we'll go back to, we'll just kind of like try to think what happened or what we learned in, xenopus, blastula formation or something, until they form the blastula, and what kind of cell division they will have, what kind of cell cycle they will have. Did I talk about it, or I never talked about it? So my question is that, okay, they are like a cell cycle, cell division heavy that period of time, isn't it? A 2-cell stage, 4-cell stage, 8-cell stage, and then 16-cell stage. They just keep on going. During that time, do they have the same cell cycle that we learned from here? Something that we learned.
The cell
Probably
doesn't
no.
go in between division.
Probably not, right? Yeah. So what do you think? What kind of cell cycle do they have? During the cleavage division, early division, okay? Okay, so I'm going to talk about it later, but I'll just give you the answer and then I'm gonna move on. They don't have G phase during that time. Okay, so they only have S and M, and S and M, okay? So think about it. If they have, let's say what we call is an abbreviated cell cycle. So if cell cycle has only S and M, then what will happen? As they go through the S and M, just keep on going like that, then what will happen to the cell?
They get smaller.
Yeah, it gets smaller, isn't it? So they cannot just keep on going forever, because later on, if they just keep on going the same way, then probably they'll run out of the cytoplasm. So probably they cannot, divide anymore. So mostly they will do it up to that point, and then later on they have to gain some kind of other phase. They have to stop for a while to produce something. Okay, right. Okay, so I'll move on. Okay, so cell has to also decide to kind of moment that need to divide. So that is really important thing, is that cell has to reach a certain size and then they will divide. And then also whether they finish their replication or something, that is also very, very important factor in determining this is the time that they have to divide. Okay. So also the timing is very, very important. I think this is the slide that I talked about already. This oocyte turning it into oocyte before the fertilization. This is like a meiosis. Somehow they started on mitosis and then they become meiosis, right? And then right before the fertilization, they have to keep it as a meiosis certain stage. But it takes so long, okay? It's not really as fast as the you think, as the mitosis. And after fertilized, and then they will divide. And then this division could be very, very fast. So usually from here to here, they, in case of xenopus, they divide up to 12 times, isn't it? So that division is like a synchronous division, and then they will divide like almost like a every other 30 minutes or something like that. Okay, so those timing is really important. So not every single division is like, it takes longer. This kind of like a it's a duration of the cell division is kind of like a, stretching. You can stretch it, okay, you can shorten or something. Okay, so DNA contents is really important. So cells in the G1 phase, usually they have the 1 copy number And then as they gain the more, and then cells in the G2 and M phase, usually they have the copy number is 2. So that's why they are undergoing cell division at this time. And also you can monitor this, cell fate, cell division cycle by, how we do that is that, kind of, material that, can going into DNA, and then they'll give you some kind of antibody staining or something, and then you can determine these cells are going the DNA replication or something. So I think that this is one of the example of the technique that how are we going to determine this kind of, whether, cells are dividing or undergoing replication or something. Okay. So, one of the, the reason why people, I think this is one of the example that people become really, cell cycle become a really, important subject in the cell biology is that they try to look at basically some different factors that controlling cell division. I mean, think about it. When you look at the cell division, when you learn cell division in like a high school, if you take the biology in there, or in freshman, biology or something, and then demonstration is mostly about the chromosome, isn't it? How the chromosome become condensate, become rod shape, and then they are aligned in equator, and then separate into opposite pole or something. It's very visible, and then occurs in a short period of time in a certain model. So that is very well described in the textbook, and one of the consequence from that is that people have the misconception about, hey, cell division is controlled by chromosome. Okay, so because we only thing that we looked at was a chromosome. So okay, so maybe the chromosome is the one that really controls the cell division process, because chromosome, their shape changes. But I think that later on people start to wonder, maybe there are some different factors, because without the cytoplasm they cannot really divide. I think that's what people found. So in here, you have the two population of cell, okay, and then there's a way that we can, synchronize the cell cycle. So usually you can use, I think that you will use some cancer cell. If it's not cancer cell, then they're not going to divide, right? Very well. So you have some kind of cell line or cancer cell, they are dividing all the time. And then you add some chemical to the those culture, then you can have a not 100%, but I would say about 80% or 90% of the population in the culture dish is the let's say S cell, and G1 cell, G2 cell, M phase cell. You can synchronize it. Okay, right. So after you synchronize it, and then you fuse those cells together. So you just basically mix them, and then how you fuse is that it's basically the same thing. You have let's say you have a 2 egg, and then you just crush it together, right? You can do that with the egg, right? With hand, right? But cell, what you can do is that you will use a very nasty glue. So glue these two cells together, and then it will melt down the cell membrane. Then you can force them to fuse together. It's a very a fail-prone technique or something. A lot of cell will just die off. But when you really look very hard, like a 1% of survival rate, and then when they grow, and then you can find this fused cell. So what they found is that in here you have a S phase of the cell, and then you have a G1 phase of cell, isn't it? And then when you fuse together, then G1 cell nuclei turning it into a S phase. So it was a G1s, they are arrested. So those DNA doesn't do anything, but when you fuse with the S cell, S phase cell, and then something in the cytoplasm comes to the flow over to the G1 phase and turn this G1 nucleus to the S phase. So they start to replicate their DNA. And when you do the G1 and G2, nothing happens. Okay. The interesting thing is that when you do M and S, okay, then what will happen is that N phase cycle, N phase nuclei, cytoplasm is spill over to the S phase, and then suddenly changing this S phase nuclei into N phase. So you can see very premature condensation, isn't it? Because S phase, they are undergoing replication. So DNA is like we call it a solubilized form. They are, chromatin granule that is not really thick as this, but something in here spill over and then controlling this. So suddenly S phase cells start to condensate. So that's why you can see kind of premature condensation of the S phase nuclei. Normally it's not like that, but they start to gaining kind of aggregation in the morphology. Okay, so finding out this kind of cell cycle is, of course, back then it was like I think it's early 60s through the 70s. So they find this kind of xenopus and then the cell cycle fusion experiment, and they could not find what is really going on because of the limitation in this their system. So they asked the genetic system. They use this it's a fission yeast that is a Saccharomyces pomvi, that is we call it a brewer's yeast. And then budding yeast is a Saccharomyces cerevisiae, that's a baker's yeast. And they look at the they call it a CDC, cell division cycle, okay? And then they look at those mutant. So think about it, so yeast, cell division is their reproduction, isn't it? So if you have a cell, then you create a mutation in the yeast that they cannot divide, which means that cell is gonna die, isn't it? So basically, it's a lethal mutation. You cannot, characterize the, mutants that they just die off, isn't it? So they create some kind of clever trick, is that they, use the temperature sensitivity. So they create a mutants that, okay, they will die only a certain temperature, okay? So they create a mutation in normal temperature. What is the yeast growing normal temperature? It's at 29 degrees C, isn't it? Right. And let's say if you bring that up to the 35 or something, and then they will die. But in normal temperature, they will, they will be okay. I think this is like some kind of point mutation. So that is the high temperature, or sometimes it could be low, okay? So that is, we call it a restricted temperature. And then permissive temperature is the regular temperature. So you don't see those phenotype, but only in the high or low restrictive temperature, you will see those phenotype. Okay. So that is the mutant. So I think that on your left, is a budding yeast, it's a baker's yeast. So you see this bud coming here, and then they are forming this cell, and then finally they will be pinched off, and then they'll grow for a while, and then also divide again. But in here is that this is one of the example that you can see many different type of the phenotype. And what it is that this bud, they will form the bud, and then looks like they are growing, but they could not pinch off. So they are still having the one cell structure. Okay, so I'll finish my lecture today up to here. And then you can ask me some question if you have, if I have to clarify something.
Dr. I have one question.
Sure.
So we were talking about like cells when after fertilization, they start growing, they don't follow the generic cell cycle phase. Do they still have like checks and balances if they're not doing G1 at all?
Oh, they still have the cell cycle checkpoint. Yes, so they still have the what is it? End phase checkpoints. Yes. So you can still stop your cell cycle when something is not correctly done. Is that answering your question?
Yes, it does. All right,
Okay.
Thank you.
Okay, so thank you very much.